This media was gratuitously produced by Clear Contributions Audio Video. Clear Contributions. Let's have clarity.
I'm going to look over the last homework here. There's two of these I want to look at, I think, two of these. All right, here's the one about your RNA polymerase. By the way, speaking of homework, before I forget, so a bunch of people got either nothing or something on this one, depending on how close you got. And then the other issue is I've got a bunch of people that didn't write their names on their attachments, and so when I printed them all out, I didn't know who was whose, and I had to go back and figure it out. So please write your name on your attachment. All right, so the polymerase is transcribing this piece of DNA. It's only copying one strand, and the polymerase is moving from left to right. So it's moving on the screen from left to right. The new strand it makes goes from 5 to 3. So if the new strand goes from 5 to 3, it's actually moving down the 3 to 5 strand. So here's the how I got it written up there, 5 to 3. Here's the 3 to 5. So the polymerase is moving on this strand so that it can make a new strand going 5 to 3. It's moving down 3 to 5, it makes a new strand 5 to 3. So when it does that, the new strand, if you imagine moving down this strand, will exactly equal that top strand. Moves down this strand and it creates this strand. So your answer here is G, except it has U's instead of A's instead of T's. So it'll be GU, AA, CGG, AUG, if the polymerase is moving from left to right. Now, if the polymerase is moving from right to left, it's going to be on this strand, moving from right to left, and the new strand will be made in the 5 to 3 direction. So it will match this bottom strand 5 to 3. So the new strand will be CAU, CCG, UUAC. All right. So that was confusing to probably at least half of the people here, if not more. Any questions on that one? All right. So your RNA is actually going to match one of those two strands going 5 to 3, whichever, depending on which way the polymerase is moving. It's going to just match, except for U's instead of T's. And then the other one, we'll talk about a couple more. Number 3. Some people asked in advance, and I gave them some hints on this one, and that was useful. You've got an RNA somewhere out here. There's a start codon, but you don't know where that is. All you can see is this much in the middle. Okay, so you need to translate 3 different frames. Let's just make up. And the parts you can see is A, G, U, C, U, A, G, G, C, A, C, U, G, A. And you don't know where the start codon is, so you don't know which frame is right. So the first thing you do is cut off in sets of 3s and translate this, but then assume that wasn't the right frame. So then instead cut off a different set of 3s and translate this one. And that might not be right either. So in that case, do this set of 3s and translate that one. And that's what it says where it says somewhere, indicate the amino acids encoded in the 3 reading frames. These are the 3 reading frames. This set of 3, or this set of 3s, or this set of 3s. So those are the 3 reading frames. So you're supposed to write down what you get for each of those 3 frames. And then it's in the middle of the protein, so AGU, CA, whatever, blah, blah, blah. That's CAC, let's see, this one, this one, this one, this one, ACU. So that one works, I think. The blue frame, GUC, UAG, that's a stop codon. And if it's in the middle of the protein coding, you can't have a stop codon in the middle because that's in the middle of your protein coding. You don't want to stop early. This is in the middle. It's a large protein. There's a lot left to go. You can't stop here, so that can't be right. And this one is the UCU, AGG, CAC, UGA. That's a stop codon too, so that can't be right. So the one that without the stop codon is going to be in the middle of the gene, not with a you can't have a stop codon. So that's what that meant with the 3 reading frames. All right, so some people got it. Some people asked for help, and they were got to it before it was due, and so kudos to that either way. Any questions on that one? And this last one here, there's not one necessarily. I mean, there's some. So anytime you insert or delete something, you're going to get a frame shift mutation. So frame shifts are bad. They're going to change every amino acid from that point down. And so near the end, A, that's going to give you a frame shift to the end of your coding sequence. So it's only going to affect those after it. And it's near the end, so it's not going to affect that many. B is going to do a frame shift right at the beginning, so it's probably going to affect almost all of your amino acids. C will take out an amino acid, one amino acid. D will give you deleting 4, so that's going to take out one amino acid and give you a frame shift in the middle. So that's debatable if that's worse than A, or worse than B. Although B is going to change more amino acids total, so I would argue B is worse, but I'll give you D if you want to say that as well. So and again, frame shifts are what you want to avoid the most because it's going to change every amino acid at that point going down. All right, any questions on these? All right, so now I'll try to find. I thought I had this ready to go, but I don't. I'll sort of pick up where we left off with sort of DNA techniques, and we were talking about blots, I think, last time. We talked about southern blots a little bit, mentioned northerns and westerns. We may come back to those in a minute. So the schedule here is we want to try to, Monday is a catch-up day. We probably need it. And then next test is supposed to be on Wednesday. So if we have time on Monday, we'll review stuff. Or if you have questions, bring them in. Well, we've got a few more techniques to talk about, and some of these take a little time. So we're just kind of going through in the order that they come in here in the book here. This is libraries. So there's libraries where you take DNA and kind of put a piece of DNA into a plasmid and have lots of different DNAs and lots of different plasmids, and that's a library, kind of like a library with books in it, only this is a DNA library. And there's different DNAs in your library. And we'll talk about why you would want to create a library of DNA. But first, let's talk about two different kinds of libraries. This one is a genomic library. The genome is all the DNA in your cells. So to do a genomic library, you extract the DNA. So the top figure here, you've got, in this case it's human, but it doesn't have to be human, it can be anything. You've got all the DNA. You throw some restriction enzyme into it, and it cleaves them all up into pieces. So now you've got all these pieces. And then you mix these pieces with plasmid, which has been cut with the same enzyme. So it has matching ends, and then add ligase. And the pieces basically, hopefully one piece will ligate into each plasmid. And once in a while you might get more than one, but generally it's one will ligate into each plasmid. You add ligase, and that closes the whole thing up. And now you've got random pieces of your genome in plasmids, which you can then transform into bacteria. And then you could freeze the bacteria, you could grow the bacteria, and each bacteria cell has a different random piece of the genome. So in this case, it's a human library. So each bacteria is a random piece of the human library in a plasmid. So this way you can have, it's easier to work with little pieces of the genome than the whole genome. One example of using a library. You could make a yeast library, say, for example. Let's say that you have this is, I'm sure, been done. You have a yeast cell. This is your yeast cell. And it is defective in leucine synthesis. Leukcine. So it can't make leucine. So you figure there's a mutated leucine gene in there somewhere. So here's some mutated leucine. It's got a mutation, doesn't work. And you want to actually know what that DNA is that makes leucine, what that gene is. So you have a library, and you've made this library with normal yeast that makes leucine. So everything is a good gene in there. And you basically shredded it all up with a restriction site, cloned it all into plasmids, put it into E. coli, That's how you can grow and amplify the plasmids. If you ever start to run short of library DNA, you just grow the E. coli and it makes more of them. But eventually you're going to take these plasmids out of E. coli and you'll have all these plasmids and they all have a different little piece of the genome in there. And then you're going to transform the yeast. So we transformed E. coli the other day by putting DNA in E. coli, but you can also transform yeast. So now each yeast cell has a different little plasmid in it. And each plasmid has a different random piece of this gene. So we'll just transform yeast. So we have hundreds of new yeast cells that each have a plasmid in there. And if one of these plasmids contains a piece of random DNA with the normal LU gene, then suddenly this is able to make leucine. And you can tell that because it will grow on media that doesn't have leucine in it. If you have a defective cell, it won't grow on media that doesn't have leucine in the media, in the food. But this one will grow on media without because it can make its own. Now you've actually found the gene because you've just sort of randomly put all these bits of DNA and just screened. You might look through hundreds of different yeast cells and find out which one can now grow, and that one happens to have this LU gene that was just from the library. Does that make sense, everybody? So you're just using a library to kind of break it down in little pieces and just see which piece can make up for the lack here, and that's probably the gene. Then you can go in and purify this DNA out of the cells, and then you can, grow just this plasmid only in E. coli and make new copies of that, and you'll have yourself that gene. All right, so that is one example of using a library. So the other kind of library is the cDNA library. And we've talked about this in genetics, so if anyone's like bored from this, just hang with me. What does C stand for those of you who've done cDNA libraries? No, but it sounds very much like that. It starts with a COM. It's complementary. So cDNA is complementary DNA. It's basically the other strand of DNA that matches a certain strand. So in this case, we're going to start with our cells. We're going to purify only the mRNAs. So what we're looking for is only the genes that encode proteins, as opposed to all the other RNAs. No tRNAs, no rRNAs, no small nuclear RNAs, but only the mRNAs. We purify those out, and then from the mRNAs, we'll use reverse transcriptase to make a DNA copy of the RNA. So that's what's going on. We'll talk a bit about the second step in a minute, but we'll take that blue RNA, make the DNA is now the copy that's in pink using reverse transcriptase. Now we've got a DNA copy, but what we really want is a double-strand DNA copy, so we can use a special RNase, which is a nuclease that degrades RNA only, and degrade that RNA, and then we'll just copy that DNA strand. Now we've got a double strand of DNA, and that's a cDNA. It's complementary DNA because it's made from the RNA. It's that complementary sequence, and then we've made it into a double strand. So now we have a DNA that represents each RNA. So we'll take all the RNAs, all the mRNAs out of the cell, and now we've done this process all at once in mass. We have a whole test tube full of all the mRNAs. We just do this thing, and now we've got all these different DNAs, and each DNA represents one of the original mRNAs. And so what this gives us is a collection of all the DNAs that represent RNAs that were expressed in that cell. Now, there might be genes in that cell that never were transcribed, so they never made an RNA, and you won't have a DNA for those. You only get the DNA from the genes that were transcribed, and that's because that's where you got it from the mRNA. So now you're going to have all these cDNAs, and you can also put these into a plasmid. So you have a whole collection of plasmids. You can put them into E. coli, so E. coli can basically copy them all for you and so forth. And do pretty much the same thing. So here's a nice compare and contrast between the two libraries. The genomic library, you've randomly cut your DNA. Each random piece of DNA might have a gene on it, but it might not have a gene on it. It might just have junk DNA. When you cut the DNA randomly, you might actually slice a gene in half. Sometimes, these restriction enzymes cut wherever they cut. It might be right in the middle of a gene. Your fragments might have 2 or 3 genes on them, or no genes on them. And then you put those into the library. So if I use a genomic library in this case, I might have got unlucky and had a plasmid that had 3 or 4 different genes on it. And one of those was the Lu gene. That's why this thing can now substitute for now it can grow without leucine. And it makes its own leucine. But then I've got to go back and figure out which of those 3 or 4 genes was the one that I cared about, was the one that made leucine and not something else. Whereas with the cDNA library, each little insert that I've got to put in is only one piece of RNA, because I've got individual genes that made RNAs and they made individual cDNAs. And so each plasmid only has one gene on it. And no extra DNA, no junk DNA. It's exactly one gene for each plasmid. So that's more useful in some cases, in many cases actually. Notice how if a gene was originally transcribed to a really high level and give you lots of mRNAs, you'll get more cDNAs out of it. And if one was less, you get less cDNAs out of it. So your library won't be completely balanced. It'll be rich in some cDNAs versus other ones. Yeah.
What would be a situation that the genomic library would be the advantage?
If you the one thing about the cDNAs is two things. They don't have introns in their DNA, so if you wanted to have the original gene with its introns in it for studying it or whatever, there are no introns here because at the RNA stage, the cell would have spliced the introns out. And then once you made your cDNA, there's no introns. And that's actually an advantage too, depending on if you want or don't want the introns. If you wanted E. coli to express the gene, remember E. coli doesn't deal with introns. You put a piece of DNA like this into E. coli, it won't know what to do with the introns and it won't work. This one doesn't have any introns in it because the cells already spliced those out by the time you made the cDNA, so that's good or bad. The one thing these don't have is a promoter. So in your genomic, you might have had a gene here, and it has a promoter right here, and that's where it transcribes from, and maybe your random piece cut right there, and you've got this gene in its promoter. If you've made a mRNA out of this, it starts to transcribe here. It doesn't transcribe the promoter. That's not transcribed. So you got your mRNA, then you got your cDNA that's a copy of it, and there's no promoter. So if you want this gene to actually be expressed, to be transcribed, you have to clone it. You have to paste it in. If that's your plasmid, and you put the cDNA here, there's no promoter to copy that. Now you can have a plasmid that has a little built-in promoter right here, but it's not the natural promoter. So you're not going to get some natural promoters promote really high-level transcription. Some natural ones promote low-level transcription. So if you use the genomic library, you've got the natural promoter, and it gives you the natural amount of transcription. But if you use a cDNA library, there's no promoter unless you add one, and that's going to give you whatever level that has. So it's more of an artificial situation a little bit. So that's maybe one advantage, depending on what you're trying to do. But those are all really good points. So you might want to make yourself sort of a genomic versus cDNA. This one has promoters. This one can have, but they're artificial. They're not the original promoter, but you can put one in. This one has introns, and this one no introns. This could have junk DNA. It could have multiple genes per piece. And this one just has exactly one gene per cDNA on it. So this one is more clean actually, but sometimes if you wanted the natural situation, you could have something like that. Something else I was just going to bring up, and now I can't remember what it was. I'm getting old. Yeah, if you wanted a situation that has every possible gene, then you got to use genomic, because cDNA only has the genes that were expressed in that cell and made mRNAs. And so there's a difference here. If you make your genomic library, let's say you want to make a human genomic library, for example. It doesn't matter which kind of cells you use. We all have the DNA. All the cells have all the DNA. You could use brain cells, kidney cells, muscle cells, liver cells, whatever. Grind them all up, take the DNA out, and make a genomic library. But a cDNA library will be different from different kinds of cells. So your brain cell cDNA library won't be the same as your liver cell cDNA library, because each one's transcribing different genes. there's lots of genes in a certain kind of cell. There's lots of genes in liver cells that are just never transcribed. And so if you make a liver cell cDNA library, you're not going to have every possible gene. You only have the ones that are used in liver cells. So if you want to have every possible gene, then you've got to use a genomic library. Unless you can find, I suppose you could take a brain cDNA library, liver one, skin one, kidney cell, muscles, whatever, and combine them all, and then you have everything. But even then, there might be a gene that's never expressed in any of those kind of cell types, so you wouldn't have them all. And I apologize for my messiness of a little table I tried to make here, but any other questions on the two library types? And sometimes which one you use depends on which one someone has. And so I actually used an experiment one time using a genomic library where a cDNA library probably would have worked better, but it turned out that the guy down the hall had made a yeast genomic library, and so that saved us the trouble of making anything. So we just borrowed his library, started to run out, just transferred into E. coli, copies everything, make more of it. But that's what we had, and otherwise I would have had to go and start this whole thing from scratch and make this, and I didn't do that. So it's like, well, someone already had one, that's what I used. All right, so great test question. Compare and contrast two different libraries there. All right, let's move on to PCR. How many people remember what PCR stands for? Yes, the polymerase chain reaction. And what does it do? All right, so this is a copy DNA type of thing. So we have two different DNA copying mechanisms. One of them is to transform E. coli and let E. coli do it for you. And the other one is to do this, and this is a sort of test tube copy of your DNA. Stick it in a test tube and basically stick all the enzymes into the test tube and copy it in the test tube. And there are pros and cons and different things that each thing will do. PCR has gotten better over the years. When it first came out, you could copy maybe 1,000 bases, and that's about as much as you could do and have it work. And now you can do much more, and the technology is better. But still, if you had a really large plasmid, you're better off putting it into E. coli and letting the cells copy it for you than trying to do it in a test tube. PCR, anything more than like maybe 10,000 bases or so, it's going to maybe be difficult, not work out as well. So they have their sort of advantages and disadvantages. So PCR, basically, you're going to do this in a test tube. So in your test tube, let's talk about the ingredients. You've got the DNA you want to copy, and it's usually a shorter piece. You're not trying to copy huge amounts of DNA, maybe 500, 1,000 bases or something like that. So here's the DNA, and you really, if you have like a piece of DNA, you might even put all genomic DNA, but you're really just trying to copy, say, from here to here. You're just trying to copy this little stretch. It might have some gene, or we'll talk about some other reasons why you might want to copy some little stretch of DNA, but that's the little stretch you're going to copy. Now, in order to copy DNA, you've got to first separate the two strands. How does the cell do that? How does a cell separate the two strands? I'm talking about separating, like not cutting, but like separating the two strands for replication. This is the before the polymerase. Starts with the H. The helicase. So if the cell will use helicase to separate the two strands so it can start copying one of them. In the test tube, you don't add helicase, you just heat it. So that's the other, they're both H words. So you, again, if you boil this, put it in 100-degree temperature, that'll separate the two strands. So you've got to have the two strands separated. So now my two strands are separated. And the part I want to copy is sort of between here and here. Now in the cell, once you separate the two strands, before the polymerase can actually do anything, what does it need to happen first? Or what does the polymerase need to like start copying at a certain place? It needs a primer. So in the cell, the primase enzyme puts little primers on, little RNA pieces. But in the test tube, we're going to just put artificial primers, and instead of RNA primers, they'll DNA primers, but little single-strand primers. So here's a little single-strand primer. It base pairs up to there. And here's another little single-strand primer that base pairs up to there. And so the primer is SS for single-strand DNA, and it's about in the order of 20 base pairs. Not base pair, it's not a pair, it's just bases, single-strand. About 20 bases, 18 bases, 19, 20, 25, somewhere around there, bases long. primers, you can get them on a machine. You can actually type into the machine which sequence you want, A, C, T, G, 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 A, C, whatever, and it will actually produce for you this piece of DNA that's a primer. You have to know what the sequence is right here so that it matches, and you have to know what the sequence is on here so this one matches. So you get two primers, one that kind of starts here and extends this way, and one that starts here and extends that way. And those go into the tube, and you have vast excess of primers. The machines will make, millions of copies of these things. So you've got millions and millions of primers in here, millions of each one. So you separate it by heat. You cool down till the primers stick. Too hot, of course, they won't stick because too hot. These strands separate, of course, the primers aren't going to stick, but you cool down a bit, the primers will stick, and then the polymerase kicks in and extends, grows that strand, and grows that strand. And then you repeat the process. Then you heat up again, separate the strands. Now these two strands separate, these two strands separate. You cool down, there's millions of primers, so the primers are going to stick again, and they will stick to the new strand and copy that again, and you repeat the process over and over again. So it's basically a heating, cooling, heating process. So in your tube, you've got the DNA to be copied, you've got the primers, you have the polymerase, and you have the, what else am I missing? The nucleotides. You've got to have A, C, T, and G, so you've got to have nucleotides. You add this to your tube, and then you basically put it into the machine that heats and cools and heats and cools, and 30 cycles is kind of normal. So one cycle, you copy the DNA, heat it up to separate the strands, cool down, primer stick, do another cycle, copy the DNA. Each cycle takes about 3 minutes. So you can do 30 cycles in a couple of hours, 2 or 3 hours. And it goes like, exponentially, 1, 2, 4, 8, 16, 32, 64, 128, 256, 512. By the time you're doing 30 cycles, it's like 1 billion copies of your DNA. All right, the PCR itself was invented in the mid-1980s by a guy named Kary Mullis. He was a genius, obviously, to come up with how you could do this in a test tube. Although in hindsight, it's like, well, gee, why didn't everybody figure this out? It's like one of those, this isn't that, it's not, all we're doing is heating up. Everybody knew you could heat up DNA and separate the strands, and everybody knew we already had polymerases, and all we did is just put artificial primers on here, and instead of RNA primers, we just made our own primers. That technology is not that, and just do this in a heat and cool, and it works. What's special? Let me finish my Kary Mullis thing. So he was a surfer dude. He wouldn't strike you as a like research biologist, And it reminds me very much of a guy that I went to lunch with. He was one of the biggest of bigwigs in the cell cycle field. He was a superstar. He publishes all this stuff, and he's like, he's like, in our lab we used to call him, he's like one of the gods, of cell. And he had like piercings all over his face and pierced tongue and all this stuff. Just not the kind of, not the traditional image of, your, suit and tie research, whatever. It's just. Anyway, so this guy was this surfer dude, and he invented this, got a Nobel Prize for it. Everyone does PCR now. It's just the most routine thing. Within a few years, everyone was doing it. Now, later on, he became famous again because he had this hypothesis that HIV does not cause AIDS. And he was very adamant about it. And everybody thought he was a crackpot for that, and they still do. So on the one hand, he invents PCR, gets a Nobel Prize. On the other hand, everybody thinks he's a crackpot for saying HIV doesn't cause AIDS. So that's interesting, and that's science. That's how things work. Now what's what I was going to say about PCR? Okay, what's special about the DNA polymerase we're using here? How many people have done PCR before? Anybody? One person. What's special about the DNA polymerase here? It's heat resistant. And why is that important? Because every time you're heating these things to separate the two strands, that would denature your enzymes. If you all remember, enzymes will denature it to get it too hot. That would ruin your enzyme completely by heating it. So you have to have a heat resistant polymerase in order that when you heat these up every time and separate the two strands with heat, the polymerase doesn't get killed and it still works. And that was one of the other big sort of things that he figured out that you could use as a heat resistant polymerase. And people discovered these heat-resistant polymerases because of the bacteria in like the Yellowstone hot pools. There's bacteria that live in near boiling conditions in Yellowstone, and someone got the bright idea, well, these must have heat resistant, well, all their enzymes are heat resistant, because otherwise they die. So they've got special heat resistant enzymes, including their polymerase. And so the first of these polymerases was called TAC, and that stands for Thermus aquaticus, and you don't have to know that, but that's the scientific name of these thermus, like thermal aquaticus. They live in the water, basically in hot water. That's the name of these bacteria, the scientific name of the bacteria, Thermus aquaticus. They live in hot water, and so we got the bright idea, let's take their DNA polymerase, and that's what we're going to use here. And then since then, they've discovered other kinds of bacteria that live at the bottom of the ocean with those thermal vents. There's these vents down at the bottom of the ocean where superheated water comes out from the magma inside the earth. And there's bacteria down there that have similar heat-resistant polymerases, and so that's where we get our polymerases from. Yeah.
So would those be classified as archaea?
Yes, those are archaea.
And you can cross over the genetic tools.
Yes, their polymerases still work. So that's really cool. All right, we're gonna pick up on PCR. There's a couple. I've got a little movie about animation showing you how it works, and then I've got a couple other PCR movies. The other ones I do want to talk about are DNA sequencing. There's some new technologies which I don't even know yet, but I'll talk about sort of the standard DNA sequencing technology to find out the letters, and a bunch of techniques for measuring transcription. That's kind of a big one, and we'll do that on Monday.